Hi, everyone. We'll be starting in just a moment. Okay, let's get started. Welcome, everyone. Today is our first seminar for the spring series. I'm thrilled with the lineup of our invited speakers put together for this season. So please check out the list. The schedule has been posted in our communication outlets as well as the HNRCA events website. Again, this semester we will be having a fully virtual seminar. Our format will be a speaker presentation followed by a moderated question and answer. Note, we also have the chat open, which you can use to communicate with the audience or to reach out to the panel for technical support. Our invited speaker today is Dr. Sarah Berry. Dr. Berry is Reader at King's College London in the Department of Nutritional Science. Her research focuses on precision nutrition, postprandial metabolism, food, and fat structure. She has been the academic leader for more than 30 human nutrition studies in cardiometabolic health, including the PREDICT program. So many of us are familiar with the PREDICT studies as they are fundamental in precision nutrition, which is highly relevant in the field right now, and especially of interest to us here at the HNRCA, as we are a designated clinical center as part of the NIH Nutrition for Precision Health Initiative. Therefore, we are very excited to have you talk with us about personalized nutrition. Thank you so much for being with us today. I'm now happy to introduce Dr. Sarah Berry.
Thank you, Catherine, and thank you, Tufts, for inviting me to speak, especially of your first one of 2023. So I'm going to talk about personalised nutrition for metabolic health and really try and touch on a lot of the work we've been doing over the past 4 years on the PREDICT studies at Zoe. I do need to first declare a conflict of interest that I do receive consultancy and options from Zoe, who are the tech company behind the PREDICT studies, and I'm their Chief Scientific Officer, but I'm employed only by King's College London. Okay, so I think that I would be speaking to the converted here to say that we know we've got a problem. We know that diet and lifestyle related diseases are continuing to grow globally. And this is despite really effective strategies that can reduce the risk of chronic diseases through diet and lifestyle. But despite the evidence showing the efficacy of these strategies, when we apply these at a population level, it simply isn't happening. And we need to ask ourselves the question why, and this is where personalized nutrition may come in. So firstly, do people even follow this advice? Well, we know, for example, in the UK that less than 1% of people follow our 7 UK dietary guidelines. But then there's also the question of whether the advice itself works. And I think this is particularly pertinent given that we know we all respond so differently to food. And this is where I think personalization may provide some, but not all, of the answers. Firstly, because we know, for example, if we deliver advice in a personalized way, even if it's still a generic population-based advice, but just by the personalized delivery of advice, we improve adherence to dietary guidelines and improve the diet quality of people's diet. We also know if we deliver advice that's personalized based on people's biology, so based on multi-omic measures, for example, that we can deliver more targeted advice that may be more effective in reducing the risk of chronic disease. But it's really challenging to deliver and curate personalized dietary and lifestyle advice because we are so variable. What we eat is so complicated and how we live our lives is very noisy. And we need to unravel the variability in who we are, what we eat, but also what we do and also why we make these dietary choices in order to truly deliver personalized advice. And this is really important because I think people often think about personalized nutrition simply about who you are and our biology, but we must consider all of these factors. So we know that we're incredibly complicated as individuals. We know that we have thousands of metabolic pathways, for example, that process food that may differ depending on so many different biological features, such as our genetics, such as our gut microbiome, our sex, our age, and so much more. We also know that what we eat is very complicated. We now know in nutritional research, so I'm sure all of you are aware, that it's so much more than just a simple set of nutrients that we need to consider when we think about how food impacts our health. We know that the food matrix, so the structure of the food, is really, really important in modulating the impact of nutrients within a food. We know that nutrient-nutrient interactions and non-nutrient bioactives are also key. And so we need to collect data of really high resolution, precision and breadth to be able to unravel these interactions between who we are and what we eat. And overlaid on this is the complexity of how we eat. And this is something that's really key and what we're spending a lot of time with our predict research really delving into. So by how we eat, I mean the context in which we eat our food. So the timing of our meal, the meal that we might have had earlier in the day, how much exercise we've had, how much sleep we've had, and so much more. And then lastly, obviously we have to consider, as we do with any dietary advice that we may deliver, why are we making those dietary choices? Is it due to our built environment? Is it due to our culture? You know, what are the factors that are shaping the choices that we make? And so, as well as collecting this high resolution, precision and breadth of data, I think it's really important that we collect real-time data that can assess what someone's doing at any point of time to deliver the most targeted and effective dietary advice back to individuals. Now, this simply hasn't been possible before. Why is this? Because traditionally, we've had to rely either on conducting very high quantity, but very low resolution and precision trials, such as epidemiological studies, or in research, we've had to rely on very low quantity studies, which are very high resolution and precision. So the kind of traditional randomized controlled trials that I've spent the last 20 years running before predicting maybe 20 people. Now, both of these are important and have their place, but for personalized nutrition, we need to harness the big data and novel technologies that we now have available to us. And this is why we have this really exciting time, I think, in nutritional research and this big paradigm shift in how we can conduct research that enables us to collect data at both scale, breadth, depth, and precision. And this is because we can harness digital devices, whether it's mobile phone apps, for example, or Apple Watches or Garmin's, you know, that are logging either what we're eating or how many steps we're taking or our heart rate variability and so much more. We can also use novel clinical devices such as continuous glucose monitors, and there's so many emerging now in our field. And we can use remote clinical testing where people from the comfort of their own lounge and their own armchair can provide samples that we can test to assess their biology, whether it's a DNA, a saliva sample for DNA, a poo sample for microbiome, or a dry blood spot for metabolomics. And for all of this to be possible, we need to harness citizen scientists. And I think, you know, with the COVID pandemic, people have become more and more aware of the importance of diet and the importance of lifestyle and how it might imminently affect their health. And so a really exciting time where people, I think, not everyone, but many people are interested in sharing back their information to researchers, to tech companies, in return for them receiving personalized advice and personalized information back. And by harnessing all of this, we can really unravel this complexity that I just talked about. And what this does is this allows us to go from these general population-based guidelines that we typically use to stratified, where we group people based on shared characteristics, to a more personalized approach in how we deliver advice. And this, we believe, will improve adherence, but also improve the efficacy of the advice that we're giving. And what this should enable us to do is transition from the average, such as the Eat Well guide, which is in the UK, or the My Plate in the US, to this precision nutrition plate that simply just was not possible before. Now, something that I think is really important to just pause on for a moment is this whole question around personalized versus population-based guidelines. There's a lot of discourse that many of you will be aware of, that people are either in the personalized nutrition camp or the population-based guidelines camp, or people that say personalized nutrition just simply isn't ready for prime time. Now, these are really important points that we need to address, and I'd be really happy to delve into this when we have the questions at the end. But I want to use this slide to illustrate that I don't think we should be either or. I think that we can harmoniously allow personalized and population-based guidance to sit side by side. And there's two examples from two previous studies of mine to illustrate this. So in this slide on the left, what you can see is the response. In this case, it's the postprandial lipemic, so the postprandial triglyceride response to two different types of fat. And what you see is everyone's response when you do one minus the other goes in the same direction. So each of these bars is an individual's response. And so everyone has a favorable response to the intervention. So in this case, the mean is very meaningful and population-based guidelines would be useful. So a simple example would be, let's say, fiber. We know that everyone would benefit nearly from increasing fiber in their diet. Now, this figure on the right shows another of my studies where I compared two different types of fat. And what you could see is about 40% of people had a response that favored the control and about 60% a response that favored the intervention. What this shows is in this instance, the mean actually is pretty meaningless for a large proportion of these people, because actually when we take the mean of this, there doesn't show any favorable effect of either the intervention or control. And where this is the instance where personalized nutrition would be really effective. And so I think it's really important to say that we can use personalized nutrition as an overlay to population-based advice. But we need to do this well, and there's lots and lots of considerations we need to bear in mind to deliver high quality personalized nutrition. We must consider multiple exposures, multiple responses, and multiple outcomes. Now, there are hundreds and thousands of studies that look at a single exposure and maybe multiple outcomes, or a single exposure and multiple responses. And these are really valuable to us as the research community in giving us really in-depth mechanistic data. But in order to deliver personalized advice, we need to look at the really integrated response. We need to look at all the pieces of the puzzle, because we need to get a full picture of who they are, what they're eating, why they're eating, and how they're eating. And the kind of considerations that I'd like you to bear in mind as I'm delivering this talk, and really when we're thinking about personalized nutrition, is what's causing the variability between individuals, but also really importantly within an individual. What's the genetic contribution to the specific outcome that we're interested in? If it's primarily all due to our genes, then very little is modifiable, and therefore how much power is there for people to actually modify this? And we need to think about people's morale in making these changes. The other factor, how precise are the measurement tools? We need to use really high precision measurement tools in order to deliver this precision personalized advice. And really importantly, can we collect high resolution diet data? If we're only collecting data about the macronutrients, I just don't think it's good enough. We can have two foods that have identical macronutrient compositions, but behave very differently. And I know this from my own research that I've done over the years on the food matrix. Lastly, and I really want to address this at some points in my talk, where does the how we eat, so that meal context, fit in? And let's not forget, as I mentioned earlier, why are we making those dietary choices? And this is exactly what we're doing with our PREDICT program of research. We're trying to address all of these considerations in this huge ongoing program to measure individual responses to food and lifestyle. It's a rather unique collaboration between traditional nutrition academics like myself and colleagues at other universities, including Jose Ordovez at Tufts University, and a startup tech company called Zoe, who specifically formed with the purpose of developing a personalized nutrition product. Key to all of the PREDICT research is the running of remote trials. And this is actually outdated as of a week ago. We've now completed about 48,000 people with remote clinical testing. We have a whole series of PREDICT studies, and I'm going to present mainly on this PREDICT 1 study, which is this first one that involved remote and clinic testing. But all of these other PREDICT studies that I've listed here involve exclusively remote testing. We also have a randomized controlled trial testing some of the outcomes that are related to implementing the personalized nutrition algorithms, and we have some longitudinal studies. Zoe have a website called joinzoe.com. So if you do want to find out more about all of these individual trials, please do go to there. But what I'm going to do today is just focus on this first PREDICT-1 trial. And what we aim to do with the PREDICT-1 trial, and have continued to really refine and delve into more deeply with our PREDICT-2 and our PREDICT-3 trials, is to harness our genetic, metabolomic, metagenomic, and this meal context information to predict individual responses to food, to address three key questions. How much variability is there between people and also within someone day to day? What's explaining these differences? Is it simply all due to our genetics? Is it simply all due to the meal composition? How important is the microbiome, our age, our sex, and our meal context? And then can we actually build prediction models to predict individual responses to food based on a subset of some of these characteristics? Now, the PREDICT-1 study was our first study, and this was a study that was conducted back in 2018 to 2019. It consisted of a primary cohort of 1,000 individuals that were based in the UK and were mainly twins from our Twins UK cohort. So we had about 700 twins. And then we had a validation cohort out of MGH, which was 100 individuals. And the study consisted of a very tightly controlled baseline clinic visit that was very similar to all of the many postprandial studies I've conducted over the last 20 years or so. And this consisted of a sequential test meal challenge, where what we did was we gave individuals muffins that were high in fat and carbohydrate at fasting and 4 hours later, to stress the system, to look at people's postprandial responses, so to look at their short-term immediate response to that food. And this is because we know that postprandial responses underpin many, but not all, but many of the chronic effects of food on our health. And so what we did is we had participants visit our clinic. We did various measurements of anthropometrics, body fat using DEXA, blood pressure, heart rate. They completed various questionnaires. We collected baseline samples for metagenomics, so poo samples. We did saliva swaps for genetics and metabolomics, and then we collected blood samples. We then gave these test meal challenges and collected further blood samples at regular intervals throughout the day. They then left our clinic and entered a 2-week at-home phase, and this is the 2-week at-home phase that actually is very similar to all of our ongoing PREDICT research that we're doing with these remote clinical trials. So participants were fitted with digital devices, including continuous glucose monitors. They were fitted with devices to measure physical activity and sleep. They were provided with standardized meals. These were in the form of muffins that were given in duplicate that differed in fat, protein, fiber, and carbohydrate. And so they consumed every morning, fasted, one of these meals, and they would then have them in random order on another occasion as well, so that we could look at the variability day to day within an individual in their responses. They also completed finger prick dry blood spot tests, so we could look remotely at their postprandial blood fat using triglycerides and insulin secretion using C-peptide as a surrogate. And they had access to our dietary assessment app that we built specifically for this purpose. And this involved people weighing everything they were eating, drinking, logging it all in real time. And it was monitored in real time by a team of nutritionists. And so they had a dashboard so that if I was logging that I had eaten, for example, I don't know, a burger, and I said it was a 5-kilo burger, they could in real time say, hey, Sarah, that picture that you just sent of that burger probably isn't 5 kilo, can you reweigh it? So that we could collect really accurate dietary data. People also logged their hunger, their energy, and their mood after every meal during that 2-week period. And they also completed further stool sampling. And what this enabled us to collect is a vast amount of data, but importantly, not just the amount of data and a breadth of data, but at very high resolution and precision. So we had 250,000 of these wonderful muffins consumed, which unfortunately are not particularly tasty. They were not designed to be gourmet culinary delights, but were designed to be a great vehicle to deliver quite varying nutrient composition. We've had millions of glucose readings, millions of meals logged, 60,000 triglyceride readings, for example, and lots of metagenomic and metabolomic measures. And what this does is, by collecting this amount of data at this precision and scale, really allows us to start to be able to unravel what are the predictors of the variability in response to food. I wanted to just pause before I delve into the results to say that the precision is really important, and I mentioned that at the beginning. So for nearly all of the collection tools that we've been using, we've been making sure that we're using very precise collection tools and technologies. So I illustrated, for example, with the diet data, how important this was. And then for the continuous glucose readings, there has been some discussion in the literature from some work coming out of Kevin Hall's group, questioning the accuracy of continuous glucose monitors and the repeatability, and therefore their application in precision nutrition. So we did some studies within Predict1 where we had, for example, 338 participants wearing 2 glucose monitors simultaneously on different arms to look how well they correlated, as you can see here. And we also did some work looking at how they rank foods, if it's different monitors, etc., showing that in this case, and you can go to the publication in AJCN, that we, in the case of CGM, using high precision tools. Our dietary data collection, as I said, was really important to ensure that we had the really high resolution of diet data. So we integrated barcoding, pictures, and weighed records. And what we've generated is a whole tree-like structure that maps from a brand level the types of food onto a very similar kind of tree that you have to the USDA database. So we have our level 6 foods that's now about 50,000 brand level foods recorded, so that we can look at the difference between, for example, one brand level of white sliced bread versus another brand level of white sliced bread. And what this enables us to do is really get quite deep and meaningful insights from this rich data set. Now, I'm going to use postprandial triglycerides and glucose in the clinic day for most of the rest of the presentation as a kind of the illustrative outcome of our results. So what you can see here is the change in triglycerides on the left and the change in glucose on the right, following these test meal challenges that we gave during the clinic days, that first day of the study in Predict1, where they had a high fat and carbohydrate meal at fasting at 0 hours here, and then another meal at 4 hours. And what I've done here is I've plotted the mean and the median response. Now, if we overlay to this mean and median response, which is what I've typically always reported prior to Predict in any of my dietary intervention studies, and what, if you remember, most of population-based guidelines are based on, if we overlay this with our 1,000 individuals, so each one of these grey lines is one of the 1,000 individual responses, you can see actually for many people the mean is pretty meaningless. So you can see that there's a large amount of people that sit above or below this mean line, and that also it's not in a particular direction. And we can quantify this with the CV, so you can see that the inter-individual, which is the variability between individuals, is very high for both the triglycerides and for glucose, and you can see that it's lower within an individual. So do you remember I said that each of these muffins we also gave in duplicate during the at-home phase? So what I've done here is I've calculated the variability within an individual from the at-home phase collection, just so you can see for comparison that with the variability between individuals is higher than within an individual day to day, but there's still a lot of noise day to day within an individual that we need to account for. So what's causing all of these differences? Is it all in our genes? Well, in order to look at this, we were quite fortunate because 70% of our participants were twins. And so we were able to quantify this using ACE heritability modeling. And you can see from the ACE heritability modeling how much is due to upbringing, genetics, and shared environment. And what I've done is visually tried to show you how much is due to genetics or the genetic contribution by plotting one identical twin against another identical twin. Now, if the response is all due to our genes, then you would expect quite a straight line. So we know that our height is predominantly due to our genes, and so you get this straight line, but you can see for glucose and triglycerides, it's all over the place. And what this shows to us is that genetics aren't explaining most of the differences for these particular outcomes. So what are the multiple determinants of these different outcomes? Well, there is a time in this talk to delve into each of them, but I've summarized some of the key exposures that we've been delving into, including meal composition, meal context, genetics, age, sex, microbiome, and some of the key outcomes that we've been looking at in this research. What's important to note is that, as I'm sure you would all be aware of, that the determinants differ for different outcomes. So the relative the variance explained in both the glucose and the triglyceride response by the different exposures is different. So for example, if we look at glucose on the left, meal composition of genetics and meal context, that's how we eat, explains a large amount of the variance that we see between individuals. But different exposures explain the variance for triglycerides. So for example, we see that genetics explains quite a small amount of the variance in meal context in triglycerides, and yet factors like habitual diet are a lot more important. And this is important from a personalized nutrition perspective when we think practically, what advice do we want to give back to people? What exposure do we want to focus on, depending on what outcome we want to focus on? And I think simply we can break it down to the factors that I talked about at the beginning, that it's not just what we eat, it's not just how we eat, and it's not just who we are, it's all of these three together. And what I want to do is spend the next 5 to 10 minutes really delving into meal context and this example of the kind of work we're doing, but the kind of possibilities with this rich data set that we have of really understanding the complexity of how we eat as well as who we are. So we know factors like time of day, exercise, meal ordering, sleep, eating speed, snacking, and so much more also impacts our responses to food. And I'm going to pick a couple of these out to illustrate these points. So we recently published a paper looking at sleep, and we've also more recently published work looking at alertness as well, and how sleep impacts alertness and how sleep impacts our postprandial glucose response and the relationship between the two. What we see is that poor sleep quality and also going to bed later results in a more pronounced postprandial glucose level and poorer glycemic control. So what does that mean from a practical level? Well, if I've been kept awake last night by kids or whatever, I might have a worse postprandial response today to the same meal that tomorrow, hoping I have a good night's sleep, I might have a better response to. So it really highlights the importance of that real-time personalization. We also know from our data that time of day matters that we consume. For example, our carbohydrate-rich foods. Now, this isn't anything new. There's been some great work that's come out over the years showing that on average, people that consume high carbohydrates in the morning have a lower glycemic response than if they consume them later in the day. We saw the same, as you can see here from this figure on the left. But when we started to look at the variability in this, we saw that there was a huge amount of variability where actually some people, it didn't really matter what time of day it was, and some people actually had the reverse. And we started to look, is there clusters of groups of people? And can we stratify this? Can we look based on age or based on sex or menopause status, for example? And so if we look here where we stratify according to age and we group people according to their age group, where we've got the younger ones on the left here and the older ones on the right, you can see for younger individuals, time of day seems to be hugely important. As you get older, though, time of day seems to be less important. Now, this could be simply that older individuals have poor glycemic control, and so it doesn't really matter either time of day. But again, this has really practical implications. Do we want to be giving everyone the same advice around time of day? We want to give targeted advice. We don't want people to make changes that are actually going to have quite a small impact on their health outcomes. Another result I want to show you, again, just to illustrate the kind of things that we need to be looking at when we're thinking about personalized advice, is meal order. So we conducted a sub-study as part of our Predict research called Predict Carbs, where we were looking at the impact of carbohydrate staples on postprandial glycemic responses. And as part of this sub-study, we randomly allocated people to different breakfasts, so carbohydrate staple breakfasts, so potatoes, white rice, white pasta, white bread, for example. But everyone was given exactly the same lunch meal. And what we found was that people's response to that lunch meal was somewhat determined by their breakfast meal. So if we look here with the blue line, what we found was that people, when they consumed mashed potato, they had a higher postprandial glycemic response compared to when they consumed, for example, pasta rice or white bread. And this again is just to illustrate the complexity when we're delivering personalized nutrition of thinking in that real time, what someone had as their previous meal, for example. What are they going to have as their next meal? And then some results that I presented at the RUNS conference only last month in Tokyo around snacking, I wanted to quickly mention as well. So we know that in the UK and the US, snacks account for about 20% of our energy. We therefore have a really simple dietary strategy that we could modify in order to improve health, given that most people select their snacks in isolation, i.e. they're not selecting their snacks based on maybe what other people in the family are eating, etc. But what we don't know is snacking frequency bad, or is it all about the snacking quality? And we delved into this data in our own Predict cohort, and we found that actually the frequency of snacking was not associated with any unfavorable health outcomes. Now, this is just association, it is cross-sectional analysis, so we can't be sure about the cause of association, but we found that after we controlled for multiple confounders, people that consumed more snacks did not have unfavorable glycemic control, unfavorable ASCVD risk, unfavorable blood pressure, and so much more. But what we did find was that the quality of the snacks mattered. We also found that the timing of the snacks mattered. So we found that there was 4 large dietary patterns of people in terms of their eating events, and this is shown here. So we found that there were morning snackers, afternoon snackers, evening snackers, and grazers. And when we compared people who were daytime snackers, that accounted for about 70% of our group, versus late evening, so this is after 8 in the evening, which is about 30% of our group, we did find that those people that were consuming snacks late in the evening, it is after adjusting for the diet quality, so the quality of the snacks as well as the background diet, that they had a more unfavorable glycemic control, shown from the postprandial glycemic glucose and shown from the HbA1c, and also they had a higher postprandial triglycerides. And so again, this is another way we need to think about that, how we eat, about when we're giving advice regarding snacking, in this case, for example, thinking about the time of day. Now, I can't talk about personalized nutrition without briefly touching on the microbiome. This is something we've been doing a lot of work on with our Predict studies. It isn't something that's my area of expertise. I'm an expert on postprandial metabolism. But I would be happy to take questions if you want me to delve a little bit more deeply into it. But I wanted to just give you the headline results, and we do have a paper published in Nature Medicine that delves quite deeply into this. What we were able to do with our data is identify a panel of microbiomes, or we call it a microbiome signature, that was associated with favorable or unfavorable measures of health and diet. So we were able to identify what I call very loosely good bugs, which are shown here on the left in the green circle, and this group of what I call very loosely bad bugs, shown in red. Now, these good bugs were associated with healthy measures of health and healthy measures of diet. They're also associated with lower glycemic and lipemic responses, yet the bad bugs, the converse was the case. Now, this is association data, so it is cross-sectional data, so we can't say whether there's a causal link here. And really important questions that we're now addressing with our randomized controlled trial that's due to complete next month is, can we use diet to personalize the microbiome? And can this then elicit changes in health? And these are really the really important next steps that need to occur in the field of personalized nutrition in relation to the microbiome. Something else that I want to mention as well is, we need to think very carefully about what feature of the response we should measure. And I want to do lessons from continuous glucose monitoring. And this applies really to all nutritional research, not just personalized nutrition. Now, The use of CGMs or continuous glucose monitors is exploding in healthy people, like it or loathe it. And again, there's lots of discourse around the value of this in healthy people. They are being used. And so what we need to do is we need to make sure we're using them responsibly and we're using the correct feature depending on what outcome we're interested in. But the response curve is very complicated. And if we look at the response curve that you see from a continuous glucose monitor, you can see there's lots of different features. We could look at the peak, we could look at the dip, we could look at the area under the curve, which is typically used. We could look at the increase in fasting, or we could look at the time in range. And I've, in a cartoon-like way, illustrated what we would consider based on our research favorable response in blue and unfavorable in red. And we've been doing lots of work looking at the different features. So what we found is that the peak is particularly important when we're looking at inflammation, for example, but we found that the dip is particularly important when we're interested in hunger or calorie intake as an outcome. And these findings that I'm going to show you here, we do not find when we consider either the peak or the 2-hour area under the curve as the exposure. So what we find in our PREDICT cohort, and I'm sure would be found in other healthy cohorts, is that a large proportion of people experience a dip after carbohydrate-rich foods about 2 to 4 hours after consuming them. And we have about 25% of our population that are what we call big dippers, and we have about 25% that are moderate dippers and about 50% that don't dip. So what we did was we compared individuals who have large dippers with those who are small dippers, and we looked at lots of different outcomes. And what we found was that if you separate people according to whether they're a big or little dipper, you have these big differences in hunger measured using VAS 2 to 3 hours after the meal. You find that big dippers consume their next meal before about 25 minutes before than if you're a little dipper. We find that at the next meal, when it's ad libitum, there's about 80 calories more consumed by big dippers versus small dippers. And over the day, the big dippers consume about 300 calories more than a little dipper. We've also explored day to day, if you dip one day versus not dipping another day, does this matter as well? So within participants, and this is data just hot off the press only last week. And what we find is that if we have individuals who dip one day after their breakfast but don't dip the next day, you have about 160 calorie difference in their calorie consumption over that day. And this is something we're really going to start delving into a bit more deeply. So what I want to finish up on is the big question: does personalized nutrition work? Well, we don't have an answer for this. We will, in the next month, complete our randomized controlled trial that's comparing personalized approaches using our Zoe product versus the USDA guidance. So I hope to have some answers from that. But I think there's a couple of different aspects we need to think about, and this goes back right to what I said at the beginning. Firstly, does personalized nutrition work in improving people's diet? So even if we were not delivering different dietary advice based on people's biology, does the personalization of how we deliver back information improve diet? And there's lots of evidence to show consistently, yes, it does. So if we think of the fact that less than 1% of people follow all 7 UK dietary guidelines in the UK, when we conducted studies such as Food for Me, which is a large EU study, this shows that a personalised advice does improve the diet quality and more people adhere to dietary guidelines. But does this actually improve people's health? Does changing the advice we're giving improve people's health? Well, some of you may be aware of the work coming out of Aaron Segal's group, where he developed predictive algorithms based on postprandial glycemia to predict how people would respond to different foods. He's since applied these in a chronic RCT for 6 months, where he compared the Mediterranean diet with his postprandial algorithm diet. And this was published in 2021 in Diabetes Care. And what you can see here is that the postprandial, personalized diet improved weight, improved triglycerides, and improved cholesterol to HDL ratio. there is the question raised by some people is how variable was that postprandial diet between individuals? Was it actually just putting diabetic people on a high fat diet? And so there's still important questions, I think, to answer. We hope to get some of these answers from the Preventomics program of research. This is another large EU program of research, multi-center across different European countries. And it's involved 3 key studies that are looking at personalization in different ways, whether it's about the delivery of the device, the key phenotypic characteristics around how it's personalized, and different targeted populations. Only the results from one of these studies has been published where they actually showed no benefit of personalization, but abstracts on the other 2 studies have shown some promise, with one of them showing lower BMI and the other showing improved glycemic control in the personalized arm. But I think what these studies aren't doing, and what we're trying very hard to do, is make sure that when we're running these kind of studies and when we're applying personalized nutrition, we consider the factors that I know I've mentioned throughout this presentation: the what, who, how, and the why we're making these choices. So what's the future of personalized nutrition? Well, the present state of play, whether we like it or not, is that the personalized nutrition industry is exploding. It's estimated to be between 6 to 18 billion. And the research is also increasing. As Catherine mentioned at the earlier, there's the large NIH grant that many of you may be involved in, and also our ongoing PREDICT program of research in our own RCTs. And so I think as a community, we need to make sure that we work together to harness the most precise technologies there are and learn from each other along the way. And so where to next? Well, I think that we're at this incredibly exciting time with all of these new technologies, digital devices, remote clinical testing, and citizen science. You know, I've spent 25 years in nutritional research. I've run, like Catherine said at the beginning, about 30 randomized controlled trials of postprandial studies in a very tightly controlled clinic setting. It blows my mind the speed, the pace, the breadth of data that I'm able to get by harnessing these technologies that we're using at Zoe. But I think where the future lies is in making sure we also integrate this real-time continuous advice, personalized to how people vary their life day to day. And that's why I wanted to pick up on all these kind of different examples about snacking and timing of snacking and meal ordering and sleep, and time of day, because I think it's really important that we personalize not just to someone's biology, but also to how they live their life in the way that a dietitian would in any consultation. Some really important points that we need to be very mindful of is how do we go about affecting change in people once we have all of this information? We do not want this to become the foray of the worried well or the elite and just accessible to those that can afford it. So we need to think, what are the prospects at the population level? And I think this is where stratified approaches can become very valuable, and that personalized nutrition can actually make significant advancements in enabling us to deliver stratified advice based on some very simple characteristics. And lastly, where do population-based guidelines fit? And I'll take you back to one of the slides I said right at the beginning. I do think they can sit harmoniously together. And I think it's about really understanding where broad population mean dietary advice is valuable versus individual advice. So that finishes off with the 4 words that I've said throughout, that we must consider the what we're eating, we must consider the how we're eating, we must consider the who we are, but really importantly, let's not forget the why we're eating. And that's because, in my opinion, what's the best bit about food? It's the pleasure. It's part of our culture, it's part of our emotions, and it's part of our lifestyle. And if we personalize to the extent that we remove this pleasure, then I think I would like to actually finish by saying this saying that I say in many of my talks, that if a food is too healthy to be enjoyed, it's just not healthy at all. So we must make sure that any personalization enables the pleasure of food to shine through. Lastly, I'd like to acknowledge the fabulous team that I've had the pleasure of working with on the PREDICT program research, all of the wonderful team at Zoe, Nicola Sagata at Trento, who leads all of our microbiome research, Jose Ordovas, who's at Tufts, and some other fabulous scientists at King's, MGH, Stanford and Harvard. And thank you very much for inviting me to talk today.
All right, that was an excellent presentation. Thank you, Dr. Berry. So we can take some time for questions. So for attendees, please either type your questions into the Q&A box, or if you prefer, you can use the raise hand feature, which we've enabled. We'll call on you and you can ask your question live. So first question, Amelia Bugandowski asks, thank you for a great insight. Do you think personalized nutrition be directed into more specific women's needs and addressed, for example, menstrual cycle or menopause?
Yeah, that's a really good question. And I think it's something that our work is highlighting just how little has been done, as many scientists will be aware, in females and how understudied they are, particularly in the area that we've been working. We've been doing a lot of work around menopause and looking specifically how pre, peri and postmenopausal women respond differently. And we find some really interesting differences when we look at them as a stratified group. So for example, postmenopausal women have bigger dips than their premenopausal female counterparts. What we've also been doing is lots of work where we've tried to age match pre and postmenopausal women. Obviously, menopause is an age-related transition. And so how do we know, is it simply as women get older, this happens versus the menopause itself? And so because we've got such large numbers of people, we've been able, and we actually published back in, I think it was September, in BOE Biomedicine, a paper on this where we age matched females who were pre and females who were post. We adjusted for multiple other confounders, and we saw that in age matched individuals, postmenopausal women had far greater postprandial glycemic responses to a standard meal compared to their premenopausal counterparts. Something we haven't delved into, but we want to and we plan to do, is looking at phases of the menstrual cycle. And I think that's really important. There is some studies, but not enough studies showing that responses to food, how we process foods, but also what foods we want differ, and our hunger levels according to phase of the menstrual cycle.
All right, thank you. Larissa Constantino asks, have you studied methods to deliver personalized advice in a way that's effective to change health behavior?
So we have a team at Zoe that are our user experience team, and they are continually testing different modes of how we deliver back advice, different types of advice that we deliver back on, different emphasis on different advice, to see how it improves interaction with the app, what part of the app they might interact. This is the Zoe personalized nutrition app that we have, that's very similar to the at-home phase that I talked about with Predict1. It's something we haven't studied in a formal clinical trial way, but it's something we are continually getting data from this user experience work that they do at Zoe.
Do you have hope for those methods? I guess I'm just thinking, such a low percentage follow the USDA guidelines here in the US, and you mentioned similarly for the UK. It almost sounds like those methods could be applied to any kind of diet advice. So do you have hope for those developing methods?
Yeah, I mean, I think the Food for Me program that I mentioned, and some other big programs where they actually deliver the same advice to everyone, but they deliver it in a personalized way, so people feel it's targeted, it's going to be highly effective. It shows that actually just even delivering back advice that people believe is personalized improves adherence to the dietary guidance. How we can do this at a population level across different demographics is, I think, what's really important, and where the built environment fits into this as well, and the barriers that the built environment have to that behavior change is something that is out of scope for what we've been doing at the moment, but is certainly something that's incredibly important to think about going forward. But based on the work that we've done and the customers of the Zoe product, which is where our learnings are from, the behavior change that we've had is really, really promising. But these are a highly motivated group of individuals who are paying for commercial products, obviously.
All right. Chao Lei asks, have you determined how much variation in triglyceride and glucose can be explained by gene-environment interaction? Environment being diet, mealtime, microbiome, etc.
So in our first Nature Medicine paper, we have broken down how much variance is explained by all of the different factors that we measured. And that's quantified in that first Nature Medicine paper. We've also, from the ACE heritability modeling that I showed you, can show you the, which is one of the slides earlier, show you the genetic versus the environmental upbringing. I don't know if that's what you're asking, or whether maybe the question's more related to epigenetics.
Okay. Yeah, I'm not
sorry,
sure.
if it is related to epigenetics, we actually are working with a team, again with Jose Ordovas from Tufts, actually, of researchers looking at postprandial epigenetic changes, and this is a grant from the BBSRC and EU, and we're at the final stages of that research, but I don't have the answers yet for that.
All right. Shangdang Wang asks, or says, great presentation. Could the predictive value of CGM for dippers in your healthy participants have a better predictive value as compared to patients with diabetes?
Okay, so could the predictive value of CGMs for dippers, I'm assuming that the questions being asked is there a greater association or predictive quality between dipping and our outcomes in healthy individuals compared to if we looked at dips in people with diabetes?
I think so.
And yeah, so we actually have excluded people with diabetes from our study for reasons getting it through the ethical approval. So it's something that we haven't been able to do. Be interesting to do. I think the most interesting thing I found with the work on dippers, and I know this isn't the main point of the question, is that, but I want to emphasize, is when we went and looked at the association between the typical features that are measured using CGMs, which is the 2-hour area under the curve or the peak response, we did not see the associations with those outcomes of hunger or energy intake. We only saw it when you looked at dips. And so the key point from that is to say that we need to look at different features depending on different outcomes. But I think that would be fantastic to look at, given that there's the metabolic disturbances with people with diabetes, whether we do actually see the same would be really interesting.
Okay, so another CGM question. This is from Alan Taylor. What CGM do you use? How frequently did they take a reading? And for how long did each person use the CGM?
So we've been using Abbott Freestyle Libre. We have also tested out Dexcom, which is the other main brand. The Dexcom does sampling every 5 minutes. The original Abbott Freestyle Libre was every 15, but I believe it's now every 5 minutes that it tests. They wore the continuous glucose monitor in the case of the Abbott Freestyle Libre, which is what all of our participants have used. The Dexcom we tested in a subgroup for 12 to 14 days. Now it's a 14-day use continuous glucose monitor, but obviously you need the first, you need the first day for it to calibrate, for it to settle, and then we discard the last day as well. When was it monitored? So we monitor it ourselves once the study is finished. In Predict 1, the participants were blinded to the readings. In Predict 3, which is actually the study that's embedded within the commercial product, the participants can see their readings. That's something that we have to consider in any of the analysis we're doing with Predict 3, that they're not blinded to the readings.
Okay, great. Fangfang Zhang asks, can you explain a bit more about the food logs that are using that allows instant feedback? How do they differ from traditional food logs? Will instant feedback affect how the participants choose what to eat?
Yeah, so we developed a food logging system in our Predict1 study with the ultimate aim of capturing the most detailed dietary information, and that integrated the barcoding, the pictures and weighed records. So people had the option of putting in a food and then in terms of quantifying the amount using all three or any of those 3 ways. We've since learned from that a lot, and what we have now, which is actually in the commercial product and for Predict3, is a less intensive version of that. People hate logging, it's burdensome. Anyone that's worked in dietary assessment knows that whilst it's the most important part, I think, of any dietary intervention study, it's the part that people hate the most. So we've been doing a lot of work to try and make the logging a lot more simple. It's not that dissimilar from traditional food logs in that we use drop-down menus and people can go into as much detail. So if we put in, for example, white bread, they'd then tick whether it's sliced, they'd then tick what brand it was, etc., and then the amount that they're having. Once participants have taken part in the test phase of Zoe, so we're talking about Predict3, which is the study that's embedded within the commercial product, they then get feedback based on lots of the measures that we've done during their test phase, which is where they wear a continuous glucose monitor, where they measure their stool sample, their postprandial lipemic triglyceride measures as well. They then have access to the commercial app, which enables them to log any food, and then an individual score pops up for that food based on their biological measures that we've measured. They also get instant feedback to healthier alternatives based on their own metrics. So if they particularly like a burger, for example, we might say, okay, based on your biology, but your preferences, your why that you put, what you like and how you eat, here are some healthier alternatives for you.
Okay, great. Thank you for getting into that about the feedback loop. Is kind of interesting, especially in our interventions that we do. So I think that was the last question. I'm just going to check if the panel is notifying me if there's anything I've missed. I think not. So that concludes today's seminar. Dr. Berry, thank you so much for visiting with us today. And thanks to everyone for attending. We'll see you next time. Bye.
Pleasure. Thank you, everyone.